Hello, everyone. Thank you so much for joining this session of the WDA Annual Conference. My name is Ricarda Tomlin, and I'm a research manager at Yale University, and I'm here today to talk to you about clinical trials. So what I'm going to start with is talking to you about why do we have to have clinical trials. So they have a lot of drawbacks. They're slow, they're expensive, they're a lot of work for both the clinicians and the participants and everybody else involved. They take a lot of extra time and effort beyond clinical care. So why do we do them? And these are basically our reasons. The most important one being that clinical research is really the only way to get new treatments approved by the regulatory agency and therefore on the market and available to everybody. So there's a ripple effect of benefits. There may not be a direct benefit to the participant, though there may. And in either case, whether the study ends up being successful as in proving that the treatment worked, the new treatment or not, it will always lead the field of research forward if it's set up properly. So there's, like I said, rigorous testing involved. It does help us to collect the information very systematically, which is important to make it reliable in the end. There will always be an emphasis on both safety and making sure that we figure out if the drug works or not in a pre-designed, planned fashion throughout the different stages. And in addition, clinical trials are designed to remove physician bias and to consider at what point participating of children is or is not appropriate. The guiding principles for all clinical research are these three guiding principles: respect for persons, beneficence, and justice. They guide our research and are expressed in these three basic principles of oversight by both the principal investigator, the ethics boards, and the federal agencies, as well as providing appropriate information upfront throughout the study and protecting participant welfare through those mechanisms listed here. When you enter a study, you will always be asked to provide consent in the beginning, either in a written form. If it's a non-interventional trial, sometimes it can be over the phone, but the elements have to be covered regardless, and they're listed here. So you want to make sure you have a really thorough understanding of what the study is doing, what it's changing from what you are currently doing, what the procedures are, how long you're in it, your risks and benefits, if there are any other options, what confidentiality provisions the team and the study design provide. Money should be discussed up front. So who is paying for what? Are you going to get a stipend? Is somebody going to reimburse you for extra travel, things like that? And of course, also what happens if anybody gets injured while in a protocol should be discussed up front, who your contacts are, and a big emphasis on the fact that participating in research is always voluntary, which is why you can change your mind along the way, and why consent really never stops. It doesn't stop by the time you sign the paper and you can then never change your mind. It's an ongoing process. So throughout your participation, you know, if you forget or if you have questions, and of course, if anything changes, the team will always discuss that with you. Next, I'm going to try to explain some of the terms that we use and why they are part of design of different studies. Not all of these apply to all studies, so it's just to give you a general idea of, if you come across these terms, why are they used, what do they mean. Randomization basically means that there is an unbiased assignment of participants to the different treatment groups. So for instance, if there's the new treatment and it's getting compared with a currently existing treatment, people typically are randomized through a computer algorithm or something similar that neither the participants nor the doctors have any influence over, are assigned to the different groups, so that then when we draw conclusions at the end, that they're truly random and comparable. And of course, that also ensures that everything is fair. When we're talking about blinding, we're talking about, does everybody know who's getting which treatment, or do we not know? It's rather complicated to blind studies, especially if there's two different treatments happening, but it is possible. through providing placebo, which is explained right here. So placebo just means it's a look-alike treatment that does not have any active ingredients in it. And, we often refer to it as a sugar pill, but it doesn't. There's also ways of providing IV placebos and other things like that. I'm going to go to the middle next, eligibility. What we mean by that is that there is a list of criteria in the protocol for who should and shouldn't be in the protocol. And this is mostly around safety and just the general design of the study. And we'll come back to this in a few minutes when I talk through a typical design study. And then the terms of non-inferiority versus superiority. So these are getting at what is the study trying to prove? So when we say the study is trying to prove non-inferiority of the new treatment, we're trying to prove that the new treatment is not worse than the current treatment, as compared to superiority, where we're trying to prove that the new treatment is actually better than the currently existing alternatives. And this has impact on the way the study is designed, how the outcomes are measured, and what the hypothesis going in is. Trial phases, I won't belabor because I have a separate slide about those. We've already briefly mentioned IRB or ethics boards. IRB stands for Institutional Review Board. They're called ethics boards in Europe, and they're all kind of designed to make sure that there's a neutral ethics committee overseeing all research. Data monitoring boards are independent, so they're not the same members who are also designing and running the study, but they're outside consultants usually, and their job is to look at the data throughout the course of this study, especially if there are potential risks to safety, so they can look at the data before it all ends to make sure that everything looks okay and as anticipated. Not all studies have this. Expanded access is kind of a fifth option, I should say, outside of the phase 1 through phase 4 clinical trial concept, and provides access to investigational treatment outside of a clinical study. So it's not technically research, but provides access to investigational treatments that are not currently available on the open market. And then preclinical research is what we refer to the research that's done before the new treatment, medication, lab tests, whatever it may be, reaches humans. So it typically includes animal testing, general lab testing, toxicology assessment, things. Okay, and then there's different types of research. So we try to distinguish between observational versus interventional. Interventional means you get something has changed in your treatment. It could be a new medication, it could be a new algorithm that a doctor tries out, it could be a new lab test, anything like that then is used to influence care, that would all be interventional. If we just do the lab test to see how it compares to current ones, but we don't use it for clinical decision making, then that would fall in the observational bucket. And the two designs mentioned here, randomized control and crossover, I'll come back to those. Let's talk about patient registries. Many of you might be familiar since the Wilson Disease Association so generously is supporting the one that's currently conducted from Yale, but with many other participating sites, is basically designed to collect a lot of data and then have a reservoir of lots of folks collaborating to then do meaningful things with those samples, with those with that clinical data. Survey studies, record reviews, you may have been asked to fill out a survey that typically also falls in the observational types of studies. And then sample collection studies, which would be collecting your samples, but not to a particular end, but just to learn something new that does not directly impact your treatment. Okay, clinical trial phases, phase 1, 2, and 3. The main distinction is how big are they? What's the focus, and how do we kind of basically design them? So phase 1 is typically the most complex. We're really trying to establish safety here. It's often done in healthy volunteers, but as those of you who've looked into the genetic therapy trials for Wilson disease. Those do not use healthy volunteers. They use patients with Wilson disease, but they're also low enrollment, really focused on safety, lots and lots and lots of assessments, very complicated studies. And then in phase 2, if phase 1 is successful, the study would move to a phase 2 design. We typically now assign patients to groups versus in phase 1, everybody kind of gets the same treatment, often in escalating doses. The focus is now on safety, but also starting to be on efficacy, meaning how effective the treatment is. The enrollment sizes are medium, and then in a phase 3 trial, that's the one that needs to be passed in order for a medication to actually become approved by the federal agencies. So this is what we call the true test. It's the largest of the 3 groups in terms of enrollment. It focuses on the same things, it's often a slightly simpler design than a phase 2 trial, just to make it possible to enroll as many patients as you can. And as you can see, my numbers are really vague, low, medium, and high enrollment. It really all just depends on whether we're studying a rare disease or not, what the outcomes are, what the expected sensitivity of the outcome measures are. Let's say the study is cleared, phase 3, the FDA or EMA have approved the new treatment. We then sometimes go to a phase 4, which would be post-market testing. It's looking at long-term outcomes, so longer than the typical 1 or 2 year follow-ups for the other studies. And sometimes it's required by the regulatory agency, and sometimes companies or study teams do it voluntarily, just to learn more. And I'm going to show you 2 quick flow charts. This one is for a randomized control trial. So the process starts on the top left with the consent. If you agree to be in the study and you feel like everything has been explained well to you, we would then go to the screening procedures, which will determine whether you're eligible to the study or not. So that's the inclusion-exclusion criteria I discussed earlier. If you're not, your participation ends. There is sometimes a possibility to join a study at a later phase. If in phase 1 you're not eligible, it's possible that the criteria might change in phase 2 or phase 3. If you are eligible, and it is a randomized control trial, you would at this point be randomized to either some kind of experimental treatment. So that's the new treatment we're investigating versus a controlled treatment. So that's whatever treatment you're typically already on, or the one that we're comparing the 2 groups between. And then there's long-term follow-up, and then your participation ends. As compared to a crossover design, which is very similar, except that the controlled treatment is shorter and then followed by experimental treatment. And the point of this design is to give everybody a chance of experiencing the experimental treatment. So that typically makes it more palatable to patients interested in actually trying out the new treatment. And it still allows, though, for group comparison between the 2 groups. I'm going to briefly talk to you about specimen and data storage. This is just something you want to pay attention to when the study team is explaining to you. So these first 2 right here, coding versus anonymization. If they tell you that your data, your samples will be coded, that means that we replace your name and other direct identifiers with a code that allows us to then link this data to other data. So let's say we send the samples out for a lab test, we get the results back. We can then connect the results with the other background data that we have on you. It does allow us to recontact you, and it also allows you to withdraw your data or samples at a later point, as compared to anonymization, where we would send out samples completely stripped of all identifiers, and now we lose the ability to link that data. So it provides the greatest protection for confidentiality and privacy, but we do lose the ability to recontact you if we find anything while we're analyzing the data. It would no longer then have the ability for you to withdraw your data, because we don't know whose sample is which one. And it can't be linked then with other results, so it stands on its own. And there's different reasons for why sometimes one method is preferred over the other. Future research, that's typically optional for you. That means leftover samples or long-term storage of the data for purposes not currently anticipated. And then genetic testing is also typically optional, though not always. Sometimes it's part of the design. And it is it will be detailed and outlined what exactly they're going to be doing. And there are some special protections in place since genetic data is extra sensitive, since it identifies you directly. Let's talk about children in clinical research. We definitely don't think that children are mini adults, so they do need their own research and adjusted studies because they do have a different physiology. Not every study is deemed safe. Minimal risk studies are typically safe for children. If they're greater than minimal risk, there needs to be a important, finding that the research can yield in order for an ethics board to approve it. Parents and guardians will be asked to give consent. Children, if they're 7 years or older, and able to give assent, which just means they agree to be in the study. So the idea is that everybody has a voice in the conversation. The children do for themselves if they're old enough. And of course, parent and guardian has to actually give consent on behalf of their child. Let's say you are now interested in looking what kind of trials are out there, what kind of studies are out there. Here's some resources. You can certainly just ask your doctor. I think a lot of providers, especially for rare diseases, are well networked and will therefore know about ongoing studies. You should network with other patients. And then you can certainly also search the web and just make sure that the source that you're using is reliable. So clinicaltrials.gov in the US, clinicaltrialsregister.eu in the European Union. The WHO has an international website as well that tracks all trials. And then wilsondisease.org, of course, is a wonderful resource to find out about ongoing research that they know of and that are open to enrollment. And the other good thing just to know about clinicaltrials.gov, it works both as a repository for ongoing studies, but also as a source for everyone to see what research has been done in the past, were the results positive or negative. They all now get posted there. So it's a really wonderful non-commercial public database and valuable tool for both researchers and patients. And then to finish out, I just want to emphasize that all of us in the research community really know that and value that research is a partnership between the investigators and the participants. So researchers need to protect your rights and your welfare, but we're also here to ensure that the data is of high quality and it is reliable. And so both of those goals rely on a partnership with the participants. We want participants to feel actively engaged in the research. And of course, there's also the piece that the physician researchers often, though not always, collaborate with private companies. And then here are the parts that make your participation active. So we want you to actively engage during the consent, ask lots of questions, make sure you really understand what we're asking you to do up front, but to ask follow-up questions. It's really important for everybody to provide accurate information, for us to provide accurate information to you, and for you to provide accurate information to us. If you're not feeling well, whether you think it's related to the study or not, you let us know about it so we can capture all those issues, come to your visits, follow the medication instructions if it is an interventional study, and call us before you start new medications to avoid drug-to-drug interactions. So that's pretty much it. This is my summary right here. The most important thing is that research participation is indeed voluntary. You have lots of rights, and so make sure you ask questions up front and throughout the course of this study. We know that it does take extra time and effort, and we're so appreciative of anybody who is willing to make the decision to participate, but if people can't, we understand. It is a way to pay it forward for future patients and developments in the field. And we talked about both the databases and the collaborative effort. And that's it. And here's my contact information in case I don't get to answer all the questions and you have a burning question, let me know. And you can reach me this way. Thank you so much.